Did you hear what happened in Tremblant?
Oh my god, Tremblant was a crazy time.
I just can't believe that. Like, why would that happen?
I don't know where she would get the nerve to do something.
Okay, girls.
Like, no, it's his fault.
Girls. Settle down. Stop talking about Tremblant. It was a week ago. Get over it.
What did you do this Mr. Break, Mr. Letteridge?
Okay. You guys can come back for free association at 11:40.
That's lunch.
Some kind of sick joke.
Okay. So today is the agenda. Joke of the day. It's a good one, ladies and gentlemen.
Aren't they all?
After, we're going to watch a brief video on protein synthesis.
Oh my god, they're so
Boring.
old.
Hey!
Boring, guys.
Maybe someday you can do it better.
Oh my god.
Foreshadowing.
And after that, at 11:40, you guys could have your free association right here in this room.
So some kind of sick joke? That's lunchtime.
I know, he thinks he's so funny.
Alright, on to the joke of the day. Joke of the day, it's a good one. Okay, so believe it or not, guys, I, Mr. Letteridge,
Oh, Mr. Letteridge.
was a bartender back in my day.
I can tell by the earring.
So, a neutron walked into the bar, sat down, and asked for a drink. Gave it to him. When he was done, he said, "So, how much do I owe ya?" And I said, "Oh, don't worry. For you, no charge."
Oh!
You guys get it?
So funny.
Yeah.
Better than yesterday's.
Yeah.
Neutrons don't have a charge, you get it? Oh, correct myself.
Alright,
now, onto the video on protein synthesis. Pay attention. We're going to have to do a project on it someday. Okay, and now for the great video on protein synthesis. Here we go.
Protein synthesis. Synthesis of protein. It's a process that sustains life, as it enables the formation of proteins, which are very important components of our bodies. Proteins are the most diverse molecules and among the most important. Their function include biological catalysts, protection against disease, and the transportation of important substances throughout the body. There are also structural proteins, such as keratin found in fingernails and hair. The first step to protein synthesis is transcription. Transcription occurs when the DNA is transcribed into messenger RNA or mRNA. The mRNA's job is to transport the message encoded in DNA to the cytoplasm, where it will bind with the ribosome. 3 steps. Transcription occurs in 3 steps: initiation, elongation, and termination. During the initiation process, RNA polymerase binds to the DNA at a promoter region. The DNA is unwound, which in turn exposes the template strand. Then, elongation commences. The mRNA is synthesized using the template strand of DNA in the direction of 5' to 3'. The mRNA will be complementary in terms of bases to the template strand, possessing uracil as a base instead of thymine. This will make it almost identical to the gene component of the DNA. RNA polymerase, the enzyme responsible for transcription, moves along the template strand, creating the mRNA with complementary bases. After being transcribed, the DNA rewinds into a double helix. RNA polymerase reaches termination when it arrives at the end of the gene sequence. At this point, RNA synthesis comes to an end, and the RNA polymerase releases itself from the DNA strand. The newly formed mRNA then leaves the nucleus and enters the cytoplasm, heading for the ribosomes.
The second step to protein synthesis is translation.
But wait! Before the mRNA can go into the cytoplasm to the ribosome, it must undergo a few changes. First, it must add a cap of GTP, guanine nucleoside triphosphate. After that, the introns are removed by a special enzyme called spliceosome. Introns are the non-coding part of the gene, therefore useless. And finally, a poly-A tail is added on. Now the mRNA is ready to go take on the ribosome.
The large ribosome contains 2 tRNA binding sites, the acceptor site A and the peptide site P.
Yeah, you did it wrong.
The large ribosome contains 2 tRNA binding sites, the acceptor site A and the peptide site P. The tRNA, transfer RNA, delivers the amino acids necessary to the ribosome. The P site holds the tRNA that is attached to the growing polypeptide chain. The A site carries the next amino acid to be added to the chain. As mentioned earlier, tRNA has the important task of transporting amino acids to the ribosomes. tRNA is L-shaped. The 3 unlinked nitrogen bases at the end loop is called the anticodon. It binds with a specific codon on the mRNA. The 3 unlinked bases at the open end of the chain is the acceptor codon, and it binds a specific amino acid. The ribosome moves along the mRNA, reading the information or code contained in the codons. Translation does not occur until the ribosome reaches the codon AUG, which is the start codon, and corresponds to the amino acid methionine. When the start codon is in the P site, a tRNA delivers the amino acid methionine. The anticodon is able to recognize which amino acid to bring because it is complementary to the mRNA. The second codon is now in the A site. The appropriate amino acid is delivered to the site, and a peptide bond is formed between the 2 amino acids. The ribosome then shifts over the next codon, and the tRNA is released, allowing it to retrieve the same amino acid for the next time it is needed. Another tRNA delivers the amino acid that corresponds to the following codon, and the process is repeated as such. The process is continued until one of the 3 stop codons, UGA, UAG, or UAA, is read in the A site. The protein known as the release factor recognizes that the ribosome has stalled, and ribosome subunits split apart, releasing the newly formed protein and the mRNA. The protein is folded and modified, and then goes to the part of the cell where it is needed.
Looks something like this.